Hello everyone, welcome to today's webinar brought to you by Microbial Insights. This will be on science-based remediation, integrating molecular biology tools into site management. We appreciate that you are taking the time to join us today, and it is my pleasure to introduce our esteemed speaker, Dr. Debora Ramos. Dr. Ramos is a biochemist with a PhD in environmental engineering from the Federal University of Santa Catarina, with part of her doctoral research conducted at Rice University. She also completed 5 years of postdoctoral research at the University of Exeter in UK and Federal University of Santa Catarina in Brazil, focusing on the development of innovative environmental remediation technologies. Her passion for microbiology began during her undergraduate studies and has driven her career ever since. With over 17 years of experience in the environmental field, Dr. Ramos specialized in the use of molecular biology tools to assess system performances and support data-driven decision-making in remediation projects. At Worley Consulting, she provides strategic technical support for her pilot testing, full-scale remediation system design, and environmental management for industrial clients. Now, before I let Dr. Ramos take it away, I would like to remind you all, as always, that this webinar is being recorded. It will be available on the Microbial Insights website under the webinar section. If you go to the top menu at the home page, there is a webinar section for you to click on. After Dr. Ramos is done, there'll be some time for questions. So feel free to type your questions as the presentation goes on, and we'll try to read off as many as we can so that she can get you those answers. Join me in welcoming a true advocate for sustainable solutions, Dr. Debora Ramos.
Hey, thanks very much for the introduction. I must say I am very excited to be speaking here on Microbial Insights webinar. So thanks everyone for joining. So today I will be talking about science-based remediation. So basically how all of these scientific developments that were made over the years provided us with this tool, molecular biology tools, that we can integrate into our site management to be able to make more informed decisions. So starting here with the agenda. So the agenda for today includes a brief introduction about molecular biology tools and frequently asked questions about them. I will also talk about the application of MBTs in site management, so all the phases of the project where we can apply these tools and we can have valuable information in each of them. I will also share 2 case studies where we apply these tools, and we'll comment on how these tools helped us have a better understanding of the process that occurred and even allow us to be able to choose more cost-effective solutions for the site. And I will close with my perception of future trends and how we can improve the use of MBTs in field applications. So a brief introduction about Worley. So Worley is a global engineering and consulting company that is focused on sustainability and energy transition. So our global workforce includes nearly 50,000 people across 45 countries. And here in Brazil, we have offices in São Paulo, Rio de Janeiro, and Belo Horizonte. Okay, so let's start. So what are molecular biology tools? So based on the ASTM standard guide, which is a guide that talks about the application of these tools for contaminated sites, these are molecular genetic analysis that can be used to characterize and evaluate microorganisms and their relative activities. So, but why do I need them? What questions they can answer? For example, if I want to know what is the concentration of a contaminant degrader, or in other words, if critical microbes for a given process to occur are there and at which concentration. So for that, I can use qPCR analysis that allows for the quantification of target genes or target microbial population. And I can also use qPCR arrays that allow for the simultaneous detection of multiple genes and populations as well. So if I want to know is degradation occurring, then typically we use isotope technologies. So here I mentioned stable isotope probing or compound-specific isotope analysis. But if I want to know what is the community structure, then I can use next generation sequencing. So the difference here is that next generation sequencing gives me information about the entire community, while qPCR tells me about specific targets. So this one here is usually applied when for example, the degradation process are not well known, or in case I want to know more about what's going on in my site, then that would be the technology to use. And today I will emphasize only these two technologies here. I will not be talking about isotope technologies. So here, moving to frequently asked questions about MBTs, I think it's important to bring this up because these questions actually reflects the concerns that professionals have when they are considering implementing these tools in site management. So the first question is that what they can do. So as I mentioned before, these tools, they provide direct evidence of the presence and concentration of critical microbes and how they can help. So traditionally, what we do, we analyze contaminant concentrations and site geochemistry, which are absolutely essential tools that we have to use. But these tools, they do not provide the complete picture. They don't tell me if I have the right microbes there. They don't tell me at which concentrations these microorganisms are in the environment. So that missing piece of information is provided by MBTs. And how can I apply this in site management? So is that just to monitor the performance of a technology? No, actually, we can apply this in very different phases of the site management. I will explore some examples in the next slide. And also what benefits these tools provide compared to traditional tools that we normally use. They help us build a multi-evidence approach that allows us to have more confidence in the strategy we select for the site, or more confidence that the technology we implemented is actually working. So that gives us more, we are able to do more informed decisions with that. And are they commercially available? So yes, they are commercially available, and they can be readily applied for field samples. I just think it's worth mentioning that depending on where you are or where the site is located, you might have to deal with international shipping, which can be a little bit challenging. So you really need to have a good plan because samples must arrive at the lab preserved. That being said, so now I will talk a little bit about how can we apply these tools in our project lifecycle. So first, during site characterization, so here basically we are identifying the contaminants that are present. We are determining what are the constituents of interest, and we are delineating the extent of the impacts, both vertically and horizontally. Then we go ahead and develop our conceptual site model. We integrate the information we have about risk assessment, and then based on that, we conduct a preliminary evaluation of the alternatives. But if we use MBTs, we can have a better assessment of potential remediation options. So for example, we have a site where contaminant levels are high, and then we normally think of more aggressive technologies like thermal remediation or in situ chemical oxidation. because they normally provide, they are faster to degrade contaminants. And eventually we don't even think about bioremediation, but sometimes if we use these MBTs, we can maybe see if these are also options that can be considered in our assessment. And based on these results that MBTs provide us, we can even think if we can even say if a M&A it's also a solution, or if we can actually use bioremediation because we have the right organisms there. They're already there. We just didn't know. So that is some examples. And also we can see if within the site we have areas that are more promising for this kind of technologies to be employed, or if we have, if we cannot use these tools at all, because results tells us if we can or we can't consider them. So moving to remediation, and here we actually includes pre-remedial stages when we are screening for the suitable technologies. So in this step, we also normally do treatability studies. We can do pilot testing, and if the results are successful, then we move to the project design. But if we use MBTs here, we can also have a better insights into the most appropriate remedial strategy. So I can know, for example, if I'm doing a pilot test with biosparging, what I want to see is the establishment of aerobic conditions in the site, and I also want to see microorganisms shifting into aerobic ones. And I also want to understand if I have potential degraders there, and also at which concentrations they are. So these are key questions that allows me to understand which is the most effective technology and help me in the decision-making process. And for monitoring, we usually assess the performance of the technology that we applied. We can also assess the results and think of changing the strategy because, for example, the results are not as we expected, and we can even understand that we have already degraded most of the contaminant mass, and then maybe it's time to transition to monitor natural attenuation. So with MBTs, we can have an additional line of evidence that help us identify what should I do based on these results. So if I'm having, for example, a DC stall, then MBTs can tell me, is it really stall? Like, do I have the right more organisms there, and they are just at low concentrations, which means with another round of injection, I can give them a little help. Or maybe these organisms are not just there. I am adding donor and they are not being stimulated. So in this case, should I consider, for example, bioaugmentation? So that could be very helpful for us to decide what to do next. And also during closure, we can apply these tools. So for example, let's say I have applied a technology that degraded contaminants. I reduce concentrations to below site-specific target levels, but I still have some residual concentrations there. But I want to start monitoring for closure. So if I use these MBTs, they might tell me that these residual concentrations can be taken off by the microorganisms that are still there. For example, if I say that bioremediation, if I understand that bioremediation is sustained at the site, then I can have more confidence that I can actually move to a closure process. And also regulators might be more likely to approve our approach based on this line of evidence as well. So yeah, now I will share one of our case studies. The first one is about abiotic and biotic dechlorination of carbon tat and chloroform by in situ chemical reduction. So this project was developed with our partners with Jacobs, University of Toronto, and the client as well. So just a brief context about the site. The site is located in Brazil in the state of Rio de Janeiro, where a former chemical facility used to operate. And during operations, they use carbon tat and chloroform. And these compounds, they were stored in these above ground storage tanks that you're seeing here that today correspond to this area. And these activities ended up impacting the site groundwater, and remedial actions were required. So there were lots of activities at this site from previous consultancies. So there was first a human health risk assessment that was developed and identified carbon tat, chloroform, and 1122 TCA as constituents of interest. So pilot tests with molasses injections were performed both at the internal area of the site and the external area as well. And these pilot tests, they progressed to field scale systems, also with molasses injections at these areas that I'm showing here. But then this strategy, it showed a limited success, and the strategy had to be reassessed, as I will show you now. So here we have the evolution of carbon tat plume over time. As you can see at the external area here, concentrations decreased, but we saw these persistent concentrations at the internal area of the site, even after all these years. And the same was observed for chloroform plume. So we see reduction at the external area, but still concentrations at the internal area. So based on that, the client approached us and asked us to think of more efficient strategy. So a strategy that could actually deal with these concentrations, these persistent concentrations that we are seeing, and also with a strategy that we didn't have to go to the field all the time because, for example, molasses, it's a very soluble compound and it's a short-lived compound. So because of that, it required lots of injection events that disrupt site operations. So the client wants something that could be more beneficial in terms of the operation as well. So what we did, we assessed the case and understood the challenge that we have. So we had a commingled plume composed by chlorinated ethanes, and chlorinated methanes. But the big challenge here was actually the high concentrations of chloroform that we had. We had concentrations between 40 and 60 milligrams per liter of chloroform, which can be toxic to microbes and can also render bioremediation less effective, especially for the other groups of compounds that we have at the site as well. So based on that, we understood that we had to focus our remedial efforts in these groups of contaminants, because once they go away, then we could be able to degrade the remaining ones. And then the first thing we saw was how these compounds degrade. So how is their degradation pathway? So the first step here, the conversion of carbon tat to chloroform, it's mostly an abiotic process because there's very limited evidence of the biological conversion of this compound into chloroform. But then the remaining steps, so the conversion of chloroform to dichloromethane can be conducted by microorganisms. And then dichloromethane has two main pathways. The main one is the conversion to acetate, formate, hydrogen, and CO2. But as a minor pathway, it can also go to chloromethane and methane. So based on this, we wanted to combine abiotic and biotic strategies to optimize the degradation of these compounds. And for that, we used in situ chemical reduction with a product that was composed by zero valent iron. So zero valent iron could take care of this first abiotic step here. But because this product also has organic components and can also provide hydrogen for the microorganisms that work on these steps as well. And on top of that, this kind of reagent like ISCAR products, ZVI products, they offer a better longevity, which means that we don't need to go to the field all the time and inject it. So usually we inject once or twice with this kind of reagents. So that would be an added benefit. And that was the choice that we selected. So this project started in 2020, and we performed 2 injection events over a 3-year period. So the first injection event we had 15 injection points, which are these gray circles that you're seeing here. And you might be asking why they are not within the plume distribution. So that is because we were also completing the conceptual site model refinement. So we suspected there was an upgrading source around here, but we still had to wait for some decommissioning activities to finish so we can finish our investigation. And then the shape of the plume that you're seeing here is based on this CSM refinement. Then for the second phase, the injection points were distribution based on the shape of the plume, as you can see here with these red circles. But then we also collected grab samples in hush time to refine the plume and optimize the injection points. So we added another 10 injection points, which are the blue ones that you're seeing here. In terms of monitoring program, we assess conditions before and after the injections, both in phase 1 and phase 2. And for the monitoring program, we included monthly monitoring of field parameters, quarterly monitoring of geochemical parameters, COIs concentrations, and also microbial analysis. So for this project, we did qPCR for total bacteria, the Halobacter, which is microorganisms that has the ability to degrade chloroform, the associated functional genes that the Halobacter has that can act on chloroform as well. And we also conducted next generation sequencing because we wanted to have a better understanding of the process that were going on at the site. So here I will only show results from phase 2 for the sake of time and also relevance. So this process, they occur under anaerobic conditions. So the first thing we want to know is that if we were able to establish these conditions at the site. So here we have ORP results. These are the campaigns over time. And the wells, they are organized based on their location. So these are upgrading wells. These are wells close to the injection points. These are downgrading wells and wells at the external area. As you can see by the shades of red, we saw mostly negative ORP results, especially for the wells close to the injection points, which is what we expected. And we also see positive values that are these in shades of green at the upgrading wells and in the external area as well. So, okay, that is exactly what we expected. But we also monitor geochemical parameters because we want to really make sure of that. So at the wells close to the injection points, we saw reduced nitrate and sulfate concentrations, and we also saw an increase in sulfide and methane, which is also consistent with anaerobic conditions since these two here are consumed and these two here are normally produced. Okay, so based on geochemical parameters, anaerobic conditions were established. But what about microbial communities? Because at the end of the day, I am establishing these kind of conditions because I am trying to stimulate anaerobic microorganisms because they are the ones that can act on the degradation of the constituents of interest. So I also use that information to see if the injections were actually favorable to promote the growth of these microorganisms. So here I am showing two examples. So here two upgrading wells, so wells that are outside the radius of influence. And here two wells at the center of mass, so wells that are within the radius of influence. And here I'm showing results over time. I will show pie charts here. So the green ones represent aerobes, the red ones methanogens, the pink ones anaerobes, and the gray unknown or facultative microorganisms. And we can see here, baseline, we have lots of aerobes, but then after the injections, we can see that communities shifted towards anaerobic ones, as we can notice by these shades of red and pink here, especially at the center of mass. We didn't see these at the upgrading wells, so that tells me that here in this area, microbial communities were in fact influenced by these injections, and I was able to stimulate the microorganisms that would be more important for my process. In terms of results of COI's degradation, so here I'm showing carbon tat plume during baseline. We can see we had these two plumes here. 2 months after the injection, we saw only residual concentrations, and the remaining campaigns, they were below detection limits. The chloroform plume, at the baseline here, we had the high concentrations that I mentioned during the earlier slides there. So I have here 14 milligrams per liter of chloroform. Then after the injection, these concentrations decreased, but they rebounded in month 5. And then the following months, they they were below detection limits and only residual concentrations around here. Today, we are monitoring for closure, and all concentrations of these compounds are below detection limits. Here I'm also showing dichloromethane because, as I showed you earlier, this compound is produced during the degradation process. So during baseline, we obviously didn't have that. But then after the injection, we start seeing concentrations. 5 months after the injections, we saw high concentrations of that. And then in the following campaigns, they were below detection limits. But what about the degraders? I have all these MBTs that I used, and I want to see if we were able to stimulate these specific degraders and how much did I stimulated them. So here we can see that the most abundant microorganisms were actually the Helicobacter and Candidatus dichloromethanomonas, which are organisms that can degrade chloroform and dichloromethane as well. So it was very consistent to see that they were the most abundant ones here. And then based on this, we also want to see if there was a clear correlation between these organisms and these constituents of interest. So we plotted here. At the column here, we are showing chloroform and dichloromethane concentrations. And in the lines, we are plotting these two microorganisms as well as the associated functional genes that can act on this process. And what we see is that when we had high concentrations of these compounds, the microorganisms concentration was also higher. And then as they were degraded, their concentration decreased as well. So that is very consistent information that these organisms were key players participating in this degradation process. Okay, so here I'm only talking about degraders, but when we do next generation sequencing, we have so much more information. And I did want to explore that. But when we do that, after the bioinformatics steps, we receive a spreadsheet like this. So we have OTU sequences. We have here the number of reads of each sequence in my sample. And then I also have the taxonomy level. And these are long spreadsheets. It's not only here where I'm showing it goes down, down, down. So oh my God, how can I use this? Like, if you don't have experience with this, it can be a little bit scary at first, but we have some tools that can help us interpret this data and explore potential correlations, interactions between all the parameters that we are analyzing. So for this, because we wanted to integrate all of this data, geochemical, analytical, and microbial data, we used a statistic tool, which is a non-metric multi-dimensional scaling. There is an ordination method that basically indicates the similarities and dissimilarities between my samples. So every dot that you see here corresponds to the microbial community of each sample at a specific time point. And we can also plot the geochemical and analytical parameters as vectors here to understand the overall behavior of these parameters. And what we can see is that these samples, they were grouped based on their locations. And why? For example, here we have the wells that are located at the external area. And I didn't show here in this presentation, but at this area, we are doing an aerobic bioremediation at this area. And it was very consistent because they clustered close to where we had high ORP values, so very consistent with what we are doing at this area. We also had the wells located upgrading from the area where we are remediating, and they clustered here, like close to none of these samples, which is exactly what we were expecting because we didn't do anything in this area. So it makes sense they were together here in this cluster. And this chart here is for the phase 2. So during phase 2, some of the monitoring wells were not close to the injection points. So these wells here, closer to the left side of the chart, are the ones that are not within the radius of influence. And these ones are the ones that were close to where we injected the product. And they were close to wells at the center of mass after the injections. And if you see, they are all close to where these geochemical parameters are pointing to higher values of dissolved iron, methane, and TOC. And because we injected zero valent iron, we normally have an increase in dissolved iron. We have an increase in TOC levels because this product has an organic component, so it contributes to TOC concentrations, and also for methane, as methane levels also increase when we are doing this kind of process. So this one really helped us tell the story and understand what happened and how we were able to shift conditions to promote more favorable conditions, to promote the stimulation of the right microorganisms that ended up degrading our compounds. And this highlights how these MBTs helped us to have successful results. So we have a strong evidence for biological degradation of chloroform and dichloromethane. We have these favorable shifts in microbial communities. We saw a correlation between concentrations of chloroform and DCM degraders, and it also correlates with the plume shrinking as well. We saw these two were really the key players in this whole degradation process, and even the reductive dehalogenases that the Halobacter have that can act on chloroform were abundant as well. So everything very consistent, and we are moving to a site closure now. Okay, so let's go to the next case study. For this case study, we applied monitor natural attenuation as a strategy to address chlorinated ethanes at an active site. So this site here is also located in Brazil in the state of São Paulo. And based on old pictures of the site, we know that drums were stored in unpaved areas, as you can see here in this picture. And this area here corresponds to where we are seeing these chlorinated ethanes impacts. So here I'm showing tetrachloroethane plume evolution over time. So as you can see, we have considerably low concentrations of PCE here. And you can also see that the distribution of the plume didn't change much. It's mostly around this area here. And we saw the same pattern for TCE here. And we saw that for DCE. So concentrations are really close to our MCLs here. And then basically around these wells that I'm showing. So with that information, we had our conceptual site model. So as you see here, we have an upgrading well. We have a well at the border of the plume. We have these two wells here located in the center of mass, and we have two downgrading wells. We also have a river close to the property. So for us to define the strategy for this site, we looked into the human health risk assessment. So as I showed you earlier, we have low concentration of these compounds. And so there are no risks to human health. They're way orders of magnitude below SSTLs. But we do have the presence of receptors here, an ecological receptor that requires attention. So what we know about the COIs, so we know concentrations are relatively stable, and we are not seeing a potential for off-site migration because they're mostly within this area that is represented here. What we also know is that these compounds are amenable to biodegradation. Their biodegradation is very well known, and we also know a lot about which microorganisms are responsible for these degradation processes. So based on all that information, we believe that monitor natural attenuation was a suitable strategy for this site, that we didn't have to inject any amendments because the scenario is favorable for the implementation of that technology. But to assess if that was really the case, what we did is that we monitor COI's degradation over time, the byproducts degradation over time. We performed some trend analysis. We also included geochemical parameters assessments. We included microbial assessments as well. So we did qPCR and next generation sequencing. We also applied that statistic tool to better understand this data. And we performed a modeling to understand if concentrations were likely to reach the river, or if natural attenuation would be capable to prevent this off-site migration. So in terms of performance here, I am showing results based on what we are seeing here. So the well in the border of plume, the center of mass, and downgrading wells here, just for you to follow. So here we see higher concentrations of PCE at the center of mass as expected, and we can see these concentrations are reducing over time. We also saw an increase in TCE and DCE, which are PCE byproducts, so it is expected that their concentration increase when we are biodegradating these kind of compounds. We also had concentrations of vinyl chloride detected, even though they are lower, and we can see also presence of ethane concentrations as well. So based on this, I have an indication that biodegradation is occurring. And then I wanted to check the geochemical parameters as well. Do I have the anaerobic conditions, which are the conditions where these reactions occur? So ORP values, they vary a little bit, but here with this dotted line represents the average ORP values. So mostly they are close to 0. Dissolved oxygen also vary, but mostly below 1 milligram per liter. Here we have the pH with values around 6. And temperature, which here in Brazil is not really a big issue. We had concentrations between 20 and 30 degrees Celsius. So Yeah, so pretty much consistent, it tells me that we have mildly anaerobic conditions, let's say. And what about the other geochemical parameters? So we had concentrations above 1 milligram per liter of iron 2, which is expected in this sort of environment. We had concentrations below 50 milligrams per liter of sulfate, because this one is just usually consumed in this kind of environment. For TOC, except for this campaign here, we had low concentrations, so below 10 milligrams per liter mostly. But this is a site under natural attenuation, so we are not injecting anything here, not any donor, nothing like that. So we were expecting to see a little bit lower TOC levels. We had methane production and ethane production as well. So using this screening process here, we have some questions here, and based on the results that we had, some scores were assigned to each of these questions, and we ended up with 16, which tells me I have sufficient evidence of biodegradation. But do I actually have these microorganisms there? Or is it M&A continue to be efficient for this site, or will I reach a point where I have to start injecting anything, for example? So for that, we use molecular biology tools. So we monitor total bacteria to understand the overall biomass. We monitor the halococoides, which is the one who can do the complete degradation of PCE to ethene. We also monitor the functional genes, for example, TCE, which is a biomarker for the conversion of TCE to DCE, and also BVC and VCRA they are biomarkers for the conversion of vinyl and DCE to ethene. We also did next generation sequencing because we want to understand what organisms are there and their relative abundance. And based on that, we can understand the potential processes that might be ongoing in the area. So for the results, we had moderate high concentrations of total bacteria, as you can see here. For the halococoides, concentrations ranged between 10 to the third and 10 to the sixth gene copies per mil, with an average of 10 to the fifth gene copies per mil, which is a moderate high concentration, so it's good for the site. Concentrations of TCE were between 10 to the third and 10 to the fifth, so this was very consistent because at this point at the site, most of the process that are ongoing is the conversion of TCE to DCE, as we didn't see lots of vinyl chloride being produced at this moment. So that is very consistent with the fact that we had high concentrations of TCE, this biomarker here. But we also had detections of BVC and VCR, which can tell me that we have a potential for conversion of vinyl chloride to ethane. But then we also wanted to use next generation sequencing and see what else we can explore with this kind of data. So here I filtered the results. So that table that I showed you earlier, I filtered the results to only show microorganisms that had a relative abundance above 2% to make things easier to interpret. And then here I can see that I also had aerobes one, but anaerobes were also here. I also see the presence of anaerobic, these key microorganisms that are associated with chlorinated ethenes degradation, both the halococoides and the halogenemonas. They also have presence of other genera capable of reductive dechlorination, not exactly chlorinated ethenes. But then I want to see if I have this specific organohalide respiring bacteria that can act on chlorinated ethenes degradation as well. So they were all there, as I can see here. And then we also use the NMDS to understand relationships between these microorganisms and the other parameters I have about the site. And if they can tell me like a consistent story that what do I have in the source area is actually different from downgradient areas where I don't have the same COIs. So here it's the same chart. We plotted the geochemical and analytical variables. And basically what we saw is that the wells located at the center of mass, they had a cluster here, and then wells located at the downgradient wells, they cluster in a different portion of this chart here. So this information tells me that impacted areas, they have an influence over microbial communities, because that's usually the case when we have this kind of compounds. It exerts selective pressure on microbial communities, so only the ones that can tolerate these conditions may thrive, and that's what we see here. We have different microbial communities here as opposed to here. And another interesting thing is about the temporal changes. You probably may not be able to see, but these symbols here, they reflect different time points at this analysis, and we didn't see changes during over time, right? They are basically clustered together, which is different from what we saw at the other case study that I showed. But here I'm not doing anything. I'm just letting microbes grow with whatever there is in the subsurface. They are growing. They are, they're just doing what they can, and then I don't expect to see any changes. I'm not injecting everything. So that's exactly what this chart is telling me. So very consistent with all the information I have from the site and everything that we expected. So based on the concentrations we have from the helicocoides and the functional genes that are associated with this process, we understood that M&A actually represents a suitable strategy for this site, and it's being effective, and we didn't have to inject anything or do any sort of intervention, because these MBTs help support this strategy, and this was approved by regulators. And yeah, so to close the presentation today, also want to talk about my perception about future trends for us to unlock the full potential of these tools during site management. So we have some challenges here. So to deal with this data, you normally require a microbiologist or a related SME that can help you to deal with this data. And as I show you, these are large data sets that can be hard to process and interpret. So for that, as I showed you earlier in that example, we normally use a rule of thumb to filter this information based on microorganisms that show relative abundance above 2%, which is okay to do that. It helps. It gives us important information. But this may dismiss potentially important trends and information that we just are not aware of because we are not looking into that. So, but how can we do that? So I've seen some conferences and papers the application of machine learning algorithms to actually assess us in this process. So they automate data interpretation of field scale. They combine MBTs, contaminant concentrations, and geochemical data, and help us identify if there are any relevant interaction between these parameters, so that we can explore better this data and to see maybe something that we don't know, organisms that they may be helping the process. And I was just looking to the known ones, the degrader ones that we already know today. So that would be very interesting to see. And also, if I use these algorithms, I can reduce the time that I spend interpreting and treating this data and trying to extract most valuable information. So I can do this in a more efficient way with these tools. So for me, I think they could be very helpful, and that's why I think this is a very promising approach for for implementation of MBTs during site management. And with that, I think I'm ready to take questions. Thank you.
Thank you so much for that excellent talk, Dr. Ramos, and those relevant case studies were very informative. We do have a couple of questions already, but if you have not typed any questions in the question section, feel free to do so now so we can get you those answers. We'll try to read off as many as we can in that time frame, and then if you aren't unable to get your question answered, or if you have a question that pops up after the webinar is over, you can feel free to email info@microbe.com. Again, that is info@microbe.com. So the first question we have for you, we've often seen engineers use chemical data, such as like the presence of degradation products, to infer or assume microbial activity. Can you explain or provide an example of the importance of filling that missing piece of microbial data?
Yeah, sure. So that is usually what we do, right? We do COIs concentrations and we monitor geochemical parameters. But for example, sometimes we have variations in concentrations of COIs. Sometimes it's due to seasonality, and sometimes it's due to a desorption process that happened. And also, for example, for the geochemical parameters, sometimes we go to the field and we have problems with the field equipments, right? Sometimes we get this data that is hard to interpret because, for example, it doesn't make sense. And then we start questioning, is it actually working? And then we can use MBTs to really understand that, no, these variations, they can occur, but then everything is doing as planned. So at the end of the day, I need these microorganisms to degrade contaminants, and I need to know if they are there and at sufficient concentrations, and the associated things or other communities that may help. So if we have that piece of information, then we can be confident that our strategy is working, and we just need to keep monitoring, right? So I think they can be very, very valuable to help us understand if the process is occurring as it should, or if I have to maybe change the strategy.
Exactly, and that was a fantastic answer. Next one we have, while assessing the status of the groundwater plume, what effects did groundwater levels, whether it's high, average, or low, have on the results?
Well, so we normally monitor at 2 different seasons exactly to capture that variation. So we want to understand what is normal variation, so that's why we do that. And eventually, following heavy rain periods, we can see water level changing. And if I have, for example, residual concentration in the soil, sometimes it gets to the groundwater, and then it changes a little bit the pattern that I'm seeing in groundwater. But if I monitor my site with the adequate frequency, I can understand that this can eventually happen, and this isn't like a normal phenomenon. But if I have the whole picture of what's going on there, I shouldn't be worried because these variations were already expected, and we are used to them.
Exactly, variables are always a factor. Moving on to the next one, how do consultants and other industry experts help site managers who are only familiar with sort of the chemical or physical treatments sort of see the value of and adopt bioremediation approaches? And then secondly, how do we bridge that gap?
Yeah, so I think this webinar is very useful for that because I work with this data, and I see there are some barriers, and that's why I brought those frequently asked questions about MBTs, because basically they reflect these are, let's say, relatively new tools for the site management. They're not tools at all, but for the site management, they have been increasingly being applied in this for this kind of project. So I think our goal here is to actually explain the value that these tools bring, and without like getting lost in the science, if I can say that, we have to make things clear. So how these tools can help me in the decision-making process, or even, for example, for the client, I can prove that bioremediation can be a viable strategy for the site. And sometimes they're just not even considered, because there are these things that, oh, when we have high concentrations of contaminants, we can't use bioremediation. And that's not true, because as I show you in the case study, we have very high concentrations of chloroform, and chloroform is toxic to microorganisms, but that didn't seem to be a problem. So even though we applied in situ chemical reduction, the degradation of chloroform was mostly a biological process. So I think the value of the tool is this: so we can actually understand all the possibilities that we can, and we can better understand the performance, if it's working or not, and how these tools can help me make more informed decisions. So I think our goal here is to actually have a clear message for them, for those that are not as familiar with this subject as we are.
Exactly. Yeah. Making those informed decisions is sort of the concrete thing that we need. Moving on, we have a question regarding your first case study. Someone wanted to know what the average concentration of MBTs were in that first case study. And then secondly, how did you resolve the issues with passivization of ZVI?
So the MBTs, like, I think you're referring to the concentrations of the main microorganisms that we found. So they were about 10 to the 6 cells per mil, which is a moderate high range. And they were the highest one in terms of the degraders that we are looking into those samples. And when they are at sufficient concentrations, it means they're growing, and when they're growing, they're breaking bones, and so everything supports the degradation of these compounds. And can you repeat the second part, please? Because I forgot.
Yeah, totally. So how did you resolve the issues with passivization of ZVI?
We didn't have issues with that, to be honest, because as I show you, we injected twice, but during phase 1 we injected, but we knew we had an upgrading source. So we were actually degrading concentrations where we were at the area that we could target at that point. But then concentrations, we keep seeing upgrading concentrations influencing that process. And then we had to inject another one when we actually found the center of mass. And then basically it all went away. So we didn't have any issues with ZVI passivation, and we were like monitoring for how long conditions would remain anaerobic, and during all the time they remained with negative or p-values, and we keep monitoring this site, and conditions are still anaerobic. So even though we no longer have these COIs there anymore, conditions are still anaerobic. So it tells me that these conditions were there to stay.
Perfect. And once again, we'd like to thank you all from Microbial Insights. Thank you, Dr. Ramos, for presenting this and for just being a wealth of knowledge with these case studies and more about MBTs. If you all have any questions in the future, she has her contact up there in regards to the case studies, or if you have any other general questions, you can just email us again at info@microbe.com. Thank you all to everyone to listening in, and if you have anything else to add, feel free to add now. All right, so if we have nothing else to add, Dr. Ramos, I would like to thank you all for joining us today, and we wish you have a great rest of the day and to join us next month for our webinar series. Thank you all.
Sure. Thank you very much, everyone. Bye-bye.